everyone, it's Jamie Bass. I'm still alive. I don't know what came over me. I don't even have a cold. I just started coughing. Anyways, this is going to be part 4 of chapter 24, and we were talking about the gallbladder and how its role is typically misunderstood by most people. Most people think that it makes the bile. The bile is made in the liver, and that's what you have on your lobule as your hepatic duct. Those hepatic ducts aren't for lymph. I know they're green, like we're used to thinking green is lymph, but if you look back at that picture on page 886, the green is actually for collecting the bile that's made by the hepatocytes. There you go again. Maybe I'm allergic to making videos. So, let's talk about the pancreas. That's the next step in your book. Pancreas has 2 main types of cells. There's islet cells, and the islet cells are producing, they have an endocrine function. So they're producing, the insulin and the glucagon, which are important for balancing blood sugar. And then the acini cells, or acinar cells, have the digestive function, and those are releasing all of the digestive enzymes. So those islet cells are going to release the insulin and glucagon into the blood, and the acinar cells are releasing the digestive enzymes into the pancreatic duct. And you'll see on page 890 that pancreatic duct that runs through the center of the pancreas, and it merges with the common bile duct to empty into the duodenum, which is that first segment of the small intestine where the majority of the mixing of digestive enzymes occurs. So that is that is kind of the deal with the pancreas. And you'll see in that histology picture of the pancreas, the acini cells and the islet cells look very different. I'm not sure if your lab instructor is going to have you know the difference microscopically, but the islet cells tend to look like little blobs, and the acinar cells tend to look, they kind of look like little egg nests almost under the microscope. Anyways, you will see that, let's see, what other things do we need to talk about? Oh, with the pancreas. Let's go ahead and talk about pancreatic enzymes. It's in here, but I like to talk about them more specifically. So you have something called pancreatic alpha amylase, which is anything amylase, just like salivary amylase. It's going to be in charge of breaking down and digesting carbohydrates. So pancreatic alpha amylase is one of those. You also have pancreatic lipase, breaking down lipids. You have nucleases, which are breaking down nucleic acids. Most people don't realize or think about that, but if you eat an apple is made of cells, right? And so those cells have nucleic acids in them, and those nucleic acids need to be broken down by nucleases. And those nucleases are released by the pancreas. You also have proenzymes, which are released by the pancreas. These proenzymes are things like trypsinogen or chymotrypsinogen, prolactase. Those are basically enzymes to assist other enzymes. So they're little helpers. There's also proteolytic enzymes. That word proteolytic, proteo like proteins, lytic like breaking. These are enzymes that break down proteins. We had already started protein digestion in the stomach with pepsin and pepsinogen, and we're going to continue it here in the duodenum with these pancreatic proteolytic enzymes, proteases and peptidases, and they just continue to break the bonds within the protein molecules. So let's move into the large intestine. There is a little flowchart on 892 that shows you the control of pancreatic secretion, which I do think is important. It's important to understand that there you go, man. Sorry. The role of secretin and CCK. They kind of have a nice little feedback loop there involving the vagus nerve. And then we get into the large intestine. The main job of the large intestine is water reabsorption and compaction and storage of feces. And That's really, really straightforward. The layout is very similar to that of the stomach, almost. It's not like the small intestine. It doesn't have like the villi, the microvilli, and all of that. It's more pitted. And there are some secretions that go on from the large intestine, but it's mostly mucus to help lubricate the feces as it's getting prepared to be eliminated. So let's talk about the control of defecation, 896. There's a nice flow chart of that. Shows you the feedback loop. As I said, that's going to be one of those long reflexes because you're going to involve the central nervous system in that relaxation of the sphincter muscles, because you don't want to go when you're not ready to go. That would not be good. So let me specifically mention some duodenal hormones. I'm sure they were listed in your book, but just in case you like to hear me talk, some of the duodenal hormones that are important to understand are gastrin, which we've mentioned. That's also a stomach hormone, right? A gastric hormone, and that is there to improve motility and secretion, so it gets other things flowing and it gets everything moving, right? There's secretin. Secretin does what you think it does. It's there for the secretion. It increases the secretion of bile and buffers that are coming from the pancreas, and it increases motility as well. There's also something called GIP, gastric inhibitory protein, I think, and that is to basically stimulate the release of insulin to reduce blood sugar. There's CCK, cholinesister kinase. I don't know the whole word of it off the top of my head, just know it as CCK. It's there to increase the secretion of digestive enzymes from the pancreas. So CCK will be released when the intestinal phase of digestion occurs, basically to tell the pancreas to start releasing those enzymes. Enterocrinin is there to increase mucus production, and VIP, vaso and something protein, VIP is there for blood vessel dilation to get increased blood flow to the digestive system so that the mixing and churning can occur appropriately. So those are the big players there. The next part of this chapter goes through digestion, which is pretty straightforward. I mean, it's chemistry, but it's pretty straightforward. We have, of course, the digestion of carbohydrates. Carbohydrates are made of monosaccharides, right? Monosaccharide, one monosaccharide, sugar, one sugar, and disaccharides, and you put them together, you have a polysaccharide. So salivary amylase and pancreatic alpha amylase are responsible for breaking those bonds of the carbohydrate molecule and making them much, much smaller. And the glucose is actually brought in through a symport mechanism with sodium. So in other words, glucose can't come into the cell unless sodium is present. So glucose and sodium will go together, which means that glucose moves opposite of potassium, right? Because sodium and potassium always move in opposite directions. So if sodium's coming into the cell with glucose, that means potassium's leaving. Just something to consider, and that will play a role when we talk about homeostasis later. So you see the whole breakdown of everything on page 898. There's a nice little flow chart, shows you which enzyme works where and what it does. That's pretty much as basic as you need, as it gets, right? You move through the mouth, you have salivary amylase and lingual lipase, breaking down the carbs and the lipids. And then in the stomach, you have gastric lipase continuing the breakdown of fats, and you have pepsin with the proteins. And then in the small intestine, you have your amylase again from the pancreas. You have all of your lipases and then all of your proteases that are there to break down all of those large molecules. And then eventually you get to the monosaccharides, or simple sugars, right? The fatty acids and the amino acids, right? So then we have to understand how those small molecules, the monosaccharides, the fatty acids, the amino acids, how do they get into the intestinal cells to then get into the bloodstream or the lacteal? And that's what the next couple diagrams are. So on page 899, you see how that glucose is transported. You see that symport system. Step number 1, the glucose is absorbed by the symport into the intestinal cells. Boom, right there. What's going to happen? Then you also have the removal of sodium. It's going to be pumped back out because you can't just have this accumulation of sodium, and then the potassium is going to come back in, and then it's going to go out into the blood capillary, right? Although it shows it going into the red, it should be blue. It should be going into the venous system, not the arterial system. That doesn't make any sense. That's definitely a big typo in this book. Then you get to the lipid transport. Lipid transport's a little different because it has to be emulsified by the bile. So basically, the fatty acid has been created from all of the lipases, and the bile will surround that fatty acid. And the bile with the fatty acid creates this kind of bubble-like structure called a micelle. And this micelle can be absorbed by the intestinal cell. So once in the intestinal cell, proteins will attach to the fatty acid, and now it's called a chylomicron. So this chylomicron basically is like a fatty acid covered in little proteins. It can leave the intestinal cell, and it's absorbed by the lacteal going into the lymphatic system, which again meets up with the venous system, if you recall. And you see the different types of lipoproteins there on page 900. Those lipoproteins, you might be familiar with the HDLs and the LDLs if you've ever had your cholesterol checked. But that just has to do with the number of fatty acids that are on the actual structure itself. And it's important to understand how they are transported and the role that the liver plays in transporting these HDLs and LDLs, which is all described for you between page 900 and 901. Then you get to the proteins and how those guys are transported. Basically, the amino acids, which were created by the proteases, are absorbed along with the sodium, just like glucose, and then the sodium-potassium pumps the sodium back out, and it goes into the blood system. Again, it goes into the venous system, not the arterial side. Not quite sure why your book made that vessel red. That was not good. So your book then goes into water. We're receiving a lot of water through our digestive system, but we're also losing a lot of water from our digestive system. You can see ingesting about 2 liters. Your salivary glands are releasing about 1 liter. Isn't that kind of gross? 1 liter of saliva a day? Ooh, that's a lot of spit. Anyways, all of your secretions are adding water to the mixture, but then where is it going? It's all being, not all of it, the majority of it is going to be reabsorbed by your large intestine. Very small percentage is coming out at the end. Then we get to the end of the chapter, which is all about aging. Everything slows down, doesn't work right. There's a list of diseases and disorders, which you should be familiar with. That's chapter 24. So let me know if you guys have any questions, or if you want to review, or if anything I said confused you, or if you just want to say hi, that's fine. I hope you guys enjoyed chapter 24. A lot of students mistake chapter 24 for being easy, because most of us are familiar with digestion because we all eat and poop, but really it's a lot more complicated than that. So don't take it for granted. Excuse me. Read it and let me know if you need help. Alright, bye-bye.